this is Yashveen. In this video, I'll be solving paper 282, May-June 2020 9700 Biology. Question 1. Coronavirus are small viruses that are 30 nanometer in diameter. Coronaviruses are able to enter the cells of mammals and birds and can replicate within these cells. Figure 1.1 shows the entrance of a coronavirus into its host cell. A. State the key features of a virus, such as a coronavirus. So those would be a protein code and nucleic acid, which can be DNA or RNA. B. State with reasons whether a coronavirus can be seen using a light microscopes. So these are 30 nanometer in diameter, and so they are not visible because the smallest or the greatest resolution of a light microscope is 200 nanometer, and anything smaller than that is not visible because the wavelength the shortest wavelength of the light is 400 nanometers, and the resolution is half of that, which means the greatest resolution is 200 nanometers. C. With reference to figure 1.1, describe how coronavirus enters the host cell. So this is the host cell. The interior of the membrane is shown. You have phospholipids, the phospholipid bilayer, and the protein carriers which attach to the coronavirus. And these curves in the protein carriers are supposed to be complementary to the protein code so that they would be able to attach due to their shape and pull this virus inward so that the membrane folds and forms an endocytic vesicle, and that would be the entire process. Two, in a healthy mammalian heart, contraction of the four chambers is coordinated by the action of the sinoatrial node and the atrioventricular node. A. After the atria fill with blood, atrial systole occurs. State the events that lead to initiate and cause atrial systole. It is initiated by the excitation wave sent out by the sinoatrial node in the right atrium, and then the excitation wave causes contraction of cardiac muscles in the atria, and so they contract. The excitation wave spreads across the cardiac muscles, which causes contraction of muscles, that is the atrial systole. B. State and explain how the structure of heart allows the atria to contract before the ventricles. There is a gap between the impulse spreading in the atrial walls and the impulse spreading, the excitation wave spreading in the walls of ventricles, because due to a band of non-conducting fiber that does not allow impulse conduction from atria to ventricles, so the excitation wave has to pass through a band of fibers which does conduct, and that would be the atrioventricular node, which delays the impulse and then relays it, which is the reason of the fact that atria contract before ventricles. See figure 2.1 shows blood pressure changes that occur in the left ventricle, left atrium, and aorta during one cardiac cycle. E, F, G, and H are points that at which a valve opens or closes as a result of blood pressure. 1. For each of the points E, F, G, and H on figure 2.1, name the valve concerned and state whether the valve opens or closes. Blood passes down the pressure gradient, so blood would flow from areas of high blood pressure to areas of low blood pressure. Now this is the dashed line over here at the bottom represents the left atrium, and this line represents the left ventricle. And that means whenever the pressure of left atrium is greater than left ventricle, the blood would flow from the ventricle into the atrium through the atrioventricular valve. And when the pressure in left atrium decreases, it gets lower than the ventricle. That means the ventricles, the atrioventricular valves close. So the area where when pressure in left atrium is less than the pressure of left ventricle, blood would not flow from atrium to ventricle and the atrioventricular valve would close, and that would be E. Similarly, when pressure in the ventricle rises above the pressure in aorta, blood would flow from the ventricle into the aorta through the semilunar valve, and that means at F the semilunar valve would open up, and again at G the ventricle pressure in left ventricle falls below the pressure in aorta, and that means as a result of pressure, blood would flow back from aorta to ventricle, and to prevent that from happening, the semilunar valves would close. And again at H, the pressure in left ventricle falls below the pressure in left atrium, so the blood would flow down the pressure gradient from atrium to ventricle, and as a result, the atrioventricular valve would open up. Now, the atrioventricular valve in the left side of the heart would be bicuspid valve, and so you can use that instead of atrioventricular valves. To explain how Figure 2.1 provides evidence that the wall of the left atrium has a different thickness to the wall of the left ventricle. The pressure in left ventricle is caused due to the wall, the contraction in the wall. And as you can see, the increase in pressure is much higher in the ventricle than that in the left atrium. And this the greater increase in pressure is due to the fact that thicker muscle in the walls of left atrium create greater force to exert greater pressure. The Bacillus Calmette-Guerin vaccine is the only vaccine used to provide protection against the infectious bacterial disease tuberculosis. Most countries of the world have a BCG vaccination program. ATB is most commonly transmitted from person to person by aerosol infection. The causative organism is present in airborne droplets. Name the species of causative organism of TB commonly passed from person to person by aerosol infection. That would be Mycobacterium tuberculosis, whereas the Mycobacterium bovis is transmitted by drinking unpasteurized milk or by eating uncooked meat that is infected. B. In general, the countries that do not have a BCG vaccination program are high income countries that have a low number of cases of TB. In most of these countries, the vaccine is given only to babies and children at high risk of developing TB. So just one reason why a child in a country with low number of cases of disease would be at a high risk of developing TB. So that here in your answer you can write down any factor that could cause infection in the child, and that might be because the child might come into contact with an infected person. C. Countries are classified by the World Bank into one of the four income groups. Table 3.1 shows the estimated incidence of TB for 2012 to 2016 for these income groups. The incidence represents the number of new cases of TB occurring per 100,000 people in 1 year. The new cases include the number of cases that have occurred again after a period of recovery. So a relapse of a TB relapse. You have in this table you can see that as you go from low income group to higher income groups, the number of incidents decreases, and the number of incidents also decreases when you go from as the time increases, when you go from the year 2012 to 2016. You can mention those 2 points in your answer where you're supposed to describe the patterns and trends, and for the third mark you can quote some data from the table. D. There is evidence that the BCG vaccine has also provided protection against the disease leprosy. Leprosy is caused by a bacterium that is closely related to the bacteria that cause TB. Suggest why the BCG vaccine can also provide protection against leprosy. For this answer, you need to look into the mode of action of BCG vaccine. And since this is a vaccine, it is going to stimulate an immune response that would be, for example, by T cells and B cells that have a shape, that have receptors complementary in shape to the antigens on TB vaccine. So, for example, B lymphocyte. So B lymphocytes would form plasma cells. B lymphocytes with receptors complementary to antigens on TB would form plasma cells that would then secrete antibodies against the TB vaccine. And if the pathogen that causes leprosy is related to TB, that means the bacterium has antigens similar in shape to the antigens on the TB pathogen, and as a result, the antibodies synthesized against the antigens on TB can bind with the antigens on the pathogen that causes leprosy and therefore lead to the death of this bacterium. A baby can gain artificial active immunity to TB after having the BCG vaccine. A baby can also gain natural passive immunity to TB. State the differences between artificial active immunity and natural passive immunity. So, artificial active immunity would be achieved by a vaccine that would stimulate an immune response, resulting in the production of memory cells that would provide longer term immunity. Whereas natural passive immunity would be gained by feeding on breast milk that would have antibodies, because the mother would be immune to TB. And because the antigen never entered the body of the baby, no immune response was carried out, and so no memory cells were created. That means natural passive immunity is short term immunity. And so you're supposed to describe the features that distinguish the two types of immunities. 4. Collagen is a major component of the cartilage found in some of the structures of the human gas exchange system. Cells that synthesize and secrete the components of cartilage are shown as chondrocytes. Figure 4.1 is a transmission electron micrograph of a chondrocyte. With reference to figure 4.1, explain two features of the chondrocyte that show how the cell is adapted to its function. And so its function is mentioned in the question as synthesis of collagen. You'd have to look at the process of protein synthesis and its modification. The first step would be transcription, where the messenger RNA is transcribed from DNA in the nucleus. So the cell has a large nucleus to carry out the transcription half of the process, followed by translation of mRNA into the polypeptide, which would occur in the ribosomes attached to the ribosomes present on the rough endoplasmic reticulum, many of which are visible in this image. So those would be the two features which you can mention in here. B1. Describe the distribution of cartilage in the human gas exchange system. The cartilage are present in the form of C-shaped rings in trachea and in the form of plates in the bronchus, whereas they are absent from bronchioles and alveoli. To outline the function of cartilage in a human gas exchange system, so they prevent the trachea from collapsing when pressure in the trachea decreases during inhaling, and because the trachea has to move during breathing process, it also provides support. See figure 4.2 shows part of the primary structure of a collagen polypeptide. 1. Name the type of covalent bond formed between amino acids shown in figure 4.2, which would be peptide bonds. 2. Figure 4.3 shows the molecular structure of the amino acid glycine with reference to figure 4.2 and 4.3 and the function of collagen. Explain how the structure of collagen polypeptide makes it suitable for a collagen molecule. Glycine is the smallest amino acid, and that means it is also every third amino acid of each of the 3 polypeptides in a collagen molecule that form the triple helix structure, and as a result it allows closest possible packaging of the 3 polypeptides that give the collagen molecule its strength required for the many functions that it performs. There are also many hydrogen bonds that hold together the three polypeptides in a collagen molecule, which also provides strength to the molecule. 5. In most plants, sucrose is the main sugar that is transported from sources to sink. In the source, sucrose is transferred from a mesophyll cell to a phloem sieve tube through a companion cell. Describe and explain how the transfer of sucrose into a phloem sieve tube from a companion cell can lead to the transport of sugar to a sink. So you're supposed to describe the events that follow the active loading of sucrose into the phloem sieve tube element and explain those events. Once sucrose has been loaded, the water potential inside of phloem sieve tube element decreases due to solute potential, and that means water moves into the phloem sieve tube element by osmosis to raise the water potential inside. And as a result, the hydrostatic pressure at source where sucrose is just loaded increases. And because hydrostatic pressure at source is greater than the hydrostatic pressure at sink, a gradient is created, and this gradient of hydrostatic pressure is what causes the mass flow of assimilates from source to sink. B. Figure 5.1 shows part of one pathway that is used in plant cells to synthesize sucrose. The enzyme sucrose synthase catalyzes the transfer of glucose from UDPG, which is uridine diphosphate glucose, to fructose. You have a ribose molecule, uracil base, two phosphate molecules, and a glucose molecule. One, name the type of bond that is formed when sucrose is synthesized. So that would be the bond between two monosaccharides, which is a glycosidic bond. To explain why UDP can be described as a phosphorylated nucleotide. To answer this question, you'd have to look at the structure of UDP and a nucleotide. Nucleotide has a ribose, a nitrogen base, and a phosphate group. And since UDP also has a ribose, a nitrogenous base, and an extra phosphate, it is similar in structure to a nucleotide molecule. Sucrose synthase acts by using an induced fit mechanism rather than a lock and key mechanism. So that would mean the shape of active site on the enzyme is not entirely complementary to the shape of the substrate. And as a result, the active site molds around the shape of the substrate to provide a better fit. With reference to sucrose synthase and the synthesis of sucrose, outline the difference between the induced fit mechanism and lock and key mechanism of enzyme action. In your answer, you can mention a few points that are general characteristics of an induced fit mechanism and a few points that are specific to the synthesis of sucrose, and that would mean the substrate in this case are UDPG and fructose. That means they have a shape that is not complementary to sucrose, and to sucrose synthase, and so it fits around them. And so the active site of sucrose synthase molds around the substrate to provide a better fit, compared to a lock and key mechanism where the shape of substrate and active site would be complementary. CUDPG is used in some algae to synthesize a storage compound known as fluoridine starch. The molecular structure of fluoridine starch has been described as an intermediate between amylopectin and glycogen with little or no amylose. So amylopectin would be the part of starch which is branched, made of alpha glucose monomers with 1,4 and 1,6 glycosidic bonds, and glycogen would be the highly branched storage carbohydrate in animal cells. Describe the molecular structure of fluoridine starch by completing the passage. So the structure would be similar to the structures which you already know in our syllabus, and so you'd be able to answer these complete these sentences. Fluorine starch is a polysaccharide composed of dash monomers. So this monomer would be the same as that in monomer and glycogen, which is an alpha glucose molecule. The monomers are joined by 1,4 and 1,6 glycosidic linkages, one of which gives a straight chain, and the other one forms branches. I do not have the alpha symbol in my keyboard, so I'll be writing out the entire spelling. However, you can just write down the symbols in your answer to give a branching structure that is less highly branched than amylose, because amylopectin and glycogen are highly branched, whereas amylose out of those three is the least branched. 6. The mitotic cell cycle in dividing cells is very carefully controlled. Complete table 6.1 to show the correct order of stages in the mitotic cell cycle. Some of the stages have been completed for you. So you have a cytokinesis at the end, mitosis, and the stages before that. You start with the G phase, followed by S phase, which is the semi-conservative DNA replication phase, and then a G2 phase. All three of these are present in are carried out. All three of these are part of interphase of the cell cycle, whereas mitosis consists of prophase, metaphase, anaphase, and telophase. At various points during the mitotic cell cycle, checks are made. A cell goes through the cell death. If error occurs, that cannot be repaired. This makes sure that no daughter cells are produced. This makes sure that the daughter cells produced are genetically identical to each other and to the original cells. Drugs have been developed that can inhibit the mitotic cell cycle and cause the cell to carry out apoptosis. These drugs are used in the treatment of cancer. Vincristine and 5-fluorouracil are chemical compounds that act as cell cycle inhibitors and can lead to apoptosis. Vincristine binds to spindle microtubules and prevents the spindle from carrying out its function. 5-fluorouracil prevents the synthesis of thymine nucleotides. Complete figure table 6.2 to show which event in the cell cycle will occur when vincristine or 5-fluorouracil are added to healthy dividing cells at the start of the interphase stage of the cell cycle. Place a tick if the event will occur, or a cross if the event will not occur. All the boxes in the table should be completed. So vincristine binds to the spindle microtubules and prevents their action. The action of spindle microtubules starts from prophase in the mitosis, and that means S phase, which happens before mitosis, would be completed. Cell does enter prophase of mitosis because the microtubules start growing at the late prophase. That means prophase would not be completed, but it would be initiated, and the rest of the events that follow prophase would not be completed, since prophase itself would not be completed. 5-fluorouracil prevents synthesis of thymine nucleotides. The nucleotides are required during the S phase, which is the DNA replication, to build new strands, and that means S phase will not be completed, which means the rest of the events that follow S phase would not be completed either. C. Vincristine has been used in the treatment of certain types of leukemia, which is the cancer of stem cells in the bone marrow. Figure 6.1 is a photomicrograph of a blood smear of a person with one form of leukemia, which affects lymphocytes. So you have red blood cells labeled in the figure. Describe how the blood smear in figure 6.1 differs from a blood smear of a healthy person, and describe how the lymphocytes in a person with leukemia, such as those in figure 6.1, differ from those of a healthy person. So leukemia causes a production of excess production of lymphocytes that have not differentiated or matured, and as a result the normal blood composition changes, and You can make that out in the blood smear shown in the figure. There are the number of lymphocytes, which are larger than red blood cells, are shown in the figure, and that those would not be present in such a great quantity in the blood smear of a healthy person. And these lymphocytes, because they are not there because they have not properly specialized, they would not be able to carry out their functions properly. All right, we're done with this paper. Thank you for watching.